Morning, ma'am. Now, I request our faculty, Dr. Santosh Kumar, to give a welcome.
Respected guests, we welcome you again for the final session of SVCP STTP2, and we are delighted to have Sarita Madam, our guest speaker, who will be dealing with the topic Role of Clinical Pharmacist in Pharmacogenomics. Namaskar, Madam.
Hello, yeah, namaskar.
And I request Ms. Leelamrita and Ms. Padmini to give our brief introduction about Savita Madam.
With immense pleasure to introduce our today's session TL speaker, Mrs. Savita R.S., who has been associated with the College of Pharmacy, Mysore at JSS Academy of Higher Education and Research since May 2007. She is currently involved in teaching pathophysiology, pharmacotherapeutics, and clinical pharmacokinetics, apart from other courses. She has also experimented with different classroom teaching practices that allows for both student-directed as well as self-regulated learning with the support of in-house learning management system. She has attended national and international conferences where she has presented research papers in the area of pharmacoepidemiology, outcomes research, and pharmacy practice. She has worked in the area of pharmaceutical care and pharmacoeconomics in renally impaired patients for her doctoral thesis and is currently responsible for providing pharmaceutical care services in the renal unit at the University Teaching Hospital. Her other areas of interest include respiratory medicine and pediatric medicine. She is also a member of local and national professional bodies. Now I request Mrs. Savita ma'am to begin her talk.
Thank you. I'll just share my screen.
Okay.
Is it visible?
Yes, ma'am, it is visible.
Okay. So thanks for, I'd like to begin by thanking the organizers for inviting me for this talk, and I'm very grateful to you all for giving me this opportunity. The topic for today's session is the role of clinical pharmacist in pharmacogenomics. And I will just give a brief introduction. I understand that Dr. Pramod has already spoken about something about genomics in the previous session. So there might be some repetition, and I request you to bear with me. So I'll just give you a brief introduction and then tell you about the genetic variations and types, and then mention about single nucleotide polymorphisms. And I'll just give one example of a CYP450 enzyme, which has shown polymorphism. And then I'll come to the actual core of the topic, that is the roles and responsibilities of a pharmacist in providing a pharmacogenomics services. And then since I came to know that this is a large group of in this audience is our teachers, so I'll just give you a brief listing about teaching pharmacogenomics, and then some web resources for pharmacogenomics and summarize. So pharmacogenomics is a study of impact of genetic variations or genotypes of individuals on their drug response or drug metabolism. Am I audible, Vishnu? I mean, Raghu, sorry. Okay. Because your voices are very feeble. Is my voice loud enough?
Yes, ma'am, it is.
Okay.
It is audible, ma'am.
Okay. Okay. So the pharmacogenomic research is more closely related to clinical practice, and any discoveries in this field will assist the healthcare professionals, including the doctors, to determine the right drugs and the proper dose for each patient, avoid the side effects, and improve drug therapy. And some of the studies that have been done in this field have proven their utility, especially for some cardiovascular drugs, antineoplastic drugs, aromatase inhibitors, and some drugs used to treat infections. And coming to genetic variations, genetic variations will occur as either a rare defect or a polymorphism. And polymorphisms are defined as variations that occur at a frequency of at least 1% in the human population. And examples are the genes which encode for the CYP450 enzymes, mainly five of these: CYP2A6, CYP26, CYP2C9, CYP2C19, CYP2D6, and CYP3A4. So these are polymorphic and they have functional mutations of greater than 1% occurring in different ethnic groups. So functional mutations are those where the protein that encodes for a particular gene, is the production of that protein is either increased or decreased, which will affect the metabolism of the drugs. So some major types of genetic variations that can be seen are mutations, where these are changes at the level of the DNA. So one or more base pairs has undergone a change, and that change could be at random or because of an environmental factor. And there can be major deletions, insertions, and genetic rearrangements, which can affect several genes or large areas of chromosomes at once. And polymorphisms are differences in individual DNAs, which are not mutations. And the most commonly occurring polymorphism is what we call as SNPs, or single nucleotide polymorphisms. And they occur about once every 1,000 bases or so. And there are other polymorphisms like copy number variations, where some DNA repeats itself, like, uh, and there can be variation in the number of repeats. And so like nucleotide polymorphisms are seen once every 100 to 300 base pairs. And SNPs occur when one nucleotide base pair will replace another. And so this is the one which decides the differences that exist between individuals in metabolizing drugs. So when the Human Genome Project was carried out, they started assigning every DNA was mapped, and every base pair was mapped. And when they were doing that, they assigned alleles as either wild type or a variant allele. So a wild type allele was the most commonly occurring allele, and a variant allele was something which was different from that. But when they were mapping, it was found that they might find the lesser occurring allele first, but still they went ahead with keeping the name as wild type allele. So whichever was identified first was called as the wild type allele. So this is for this example is of a nucleotide sequence of the beta 2 adrenergic receptor gene from codons 13 to 19. And the nucleotide sequence with adenine in place 46, located in codon 16. And this AGA codon designates the amino acid arginine. And it occurs in about 39% of the population. And then at the same place, if the A is replaced by G, that is guanine, if adenine is replaced by guanine, that encodes for the amino acid glycine. And this occurs in about 61% of the population. So whenever you see any database, and when you're actually referring if you're doing pharmacogenomic studies, you will always have to look up the wild type allele or they would have given this as a wild type or a variant type allele. So you should be knowing what this is. So wild type is the one which was identified first when they did the genome project, and the variant allele is the variation that occurs in this allele. But need not be necessary that the wild type allele is the most commonly occurring allele. And if the single nucleotide polymorphism changes the amount or function of a protein that contributes to a drug response, then it will alter a patient's sensitivity to a drug, or it may predispose the patient to adverse reactions to drug therapy. So where do these SNPs occur? Usually they can occur in any of the three regions of the gene, either in the exon or the regulatory region or in the intron region. If a SNP occurs in the exon region, then it will alter the function of the protein. And in regulatory regions, it may alter the amount of protein that it produces. In the intron region, they might be silent. They are really silent most of the times, but they might affect the splicing of the gene. And if two or more SNPs are inherited together, then it leads to a condition called as linkage disequilibrium. And a set of SNPs which are inherited together is called as a haplotype. So coming to the cytochrome P450 enzymes, we know that about 57 different CYP450 isoenzymes are present in humans, and about 42 of them are involved in metabolism of both exogenous as well as endogenous substances. And 15 of these isoenzymes are known to involve in metabolism of drugs. And various studies have been conducted, and it is now known that there is a significant inter-individual variabilities in enzyme activities because of induction, inhibition, as well as genetic inheritance. So the example that I want to tell you about is this CYP2D6. So this enzyme, there are polymorphisms in the gene which encodes for this enzyme. So there are 100 gene variants and there are 120 alleles, and 12 SNPs are there which represent about 20 haplotypes. And you can actually test with 90 to 95% I think you might have, I do hope you know what genotype and a phenotype is. So a phenotype is the expression of genotype. You, if test predict this drug is going to be mobilized in this manner. So that is the way. And for this enzyme, there are two kinds of people. There are either extensive metabolizers or poor metabolizers. So they can be predicted with up to 99% confidence. And the gene which codes for CYP2D6 is localized on chromosome 22. And the variants, one of them is the CYP2D61, which is a wild type variant, and it exhibits normal enzyme activity. But another variant, which is the CYP2D62, this has the same activity as 2D61, but it is capable of duplication or amplification. And both these variants are present in extensive metabolizers. So poor CYP2D6 metabolizers, they carry two defective alleles. One is CYP2D63 and the more common is CYP2D64. And 2D65 and 2D66 are also there. And a total absence of an active enzyme is seen, and an impaired ability to metabolize drugs which use this enzyme for their metabolism, or they are substrates for the CYP2D6 enzyme. So some examples of CYP2D6 substrates are neuroleptic medications, mostly tricyclic antidepressants and meancerin, some antiarrhythmic drugs, and beta-adrenergic antagonists like metoprolol. So overall drug metabolism and the drug's therapeutic index the clinically significant side effects may occur in poor metabolizers because there is elevated levels of drug in the blood, and that can predispose them to an adverse drug reaction. So the example is the anti-anginal agent perhexylene, and if the drug is given to a poor metabolizer, then they are more likely to have neuropathy after treatment. Similarly, the neuroleptic agents propafenone and perfenazine, people who are poor metabolizers, they are more likely to have some Parkinson side effects when given with these drugs. And similarly, for any patient who is a poor metabolizer of CYP2D6, takes codeine or tramadol, they experience little or no analgesic effect because codeine and tramadol are converted by CYP2D6 to morphine and desmethyl tramadol. And another example is conversion of this tamoxifen, which is a drug used to treat breast cancer, that is post surgery. Uh, the drug is given for 2 to 3 years, depending upon the response. And patients who are poor metabolizers, the actual thing with tamoxifen is that it has to be converted inside the body to the active compound 4-hydroxytamoxifen. So if they are poor metabolizers, then there is a chance that they are given tamoxifen, then the breast cancer recurrence is faster, and or they might more likely, or they are more likely to progress in the to the next stages of cancer. So it would be a wrong drug to give if you know that genetically this person is a poor metabolizer of CYP2D6. And also the drug abuse is protected. So one example is if patients are addicted to morphine or any other drugs of abuse, because they are using um, this property of CYP2D6 inhibition. If you give fluoxetine, which is an inhibitor of CYP2D6 enzyme, so they metabolically, the extensive metabolizers are metabolically converted into poor metabolizers because fluoxetine inhibits this enzyme. So in such a case, you can treat effectively the drug abuse because when they take the drug, they are not getting the effects which make them to go back to take the drug. So the drug abuse is protected if you give any drug which has a potent inhibition of this enzyme. So you can use it in this way also. Then coming to the range of the CYP2D6 activity, so there are ultra rapid metabolizers on one end of the spectrum, and there are very poor metabolizers on the other end. And UMs, that is ultra rapid metabolizers, they carry a duplicated or an amplified mutant allele. So two or multiple copies of the functional CYP2D61 or the CYP2D62 allele, they have very high CYP2D6 activity. So in such patients, if you give them nortriptyline, that is an antidepressant, you will not get the required or the desired therapeutic plasma concentrations. So you will have to give 2 or 3 times the dose. So because nortriptyline has to be converted by CYP2D6 into 10-hydroxy nortriptyline. So they will require 3 times. And among both these alleles, the study showed that one of them requires 2 times the dose, and the other allele requires 3 times the dose to get the same effect. And another variant is CYP2D610. and that has been observed to be highly prevalent in the Asian population, and that is associated with lower enzyme activity. So higher drug concentrations or lower dosage requirements of neuroleptic medications and meancerin is seen in this population. And presence of CYP2D617, which is another variant in the African heritage. So they metabolize the CYP2D6 substrates at a slower rate. So, and there is a test which you can do to identify the type of variant of CYP2D6 that you have, and it is an ampli chip CYP450 test. So these are small kits that are available, and that can get you can get to know what is the variant that you are. And but the costs of treatment and genotyping are high, and especially in India, it's still high, and no doctor has advised, at least in our setup, I have not come across anybody ordering this test to identify the variant. So this is a background. I just till now I gave you a background of how knowing a knowledge of a deficiency in the metabolizing enzyme can help us to decide whether or not to give a drug and how much of dose of a drug to be given. So coming to the role of pharmacist in clinical pharmacogenomics, um, so the American Society of Health System Pharmacists, they brought out a statement for the role of pharmacist, and they said that pharmacogenomic testing will improve medication related outcomes in all health system practice settings, and this can cause a reduction in the suboptimal clinical outcomes, decreased cost of treatment, better medication adherence and appropriate selection of therapeutic agent, a decreased length of treatment, and even enhanced patient safety. So, pharmacists have a fundamental responsibility to ensure that pharmacogenomic testing is performed when needed and that results are used to optimize the therapy. And the test results will have implications throughout a patient's lifetime, and it's also a responsibility of pharmacists should have that a basic understanding of the genomics, because he is involved in providing patient care recommendations. So without the basic understanding of genomics, the pharmacist will not be able to do this. And some advanced pharmacist functions in applying clinical pharmacogenomics may require some specialized education, training, or experience. And the American Society of Health System Pharmacists, they provide
Excuse me, ma'am. The voice is not audible. We have a network issue. Ma'am will join within a couple of minutes.
Hello, madam, can you hear us? Hello?
Hello.
Yes, ma'am, we can hear you now.
Okay, it just suddenly went out, and I was just trying to
Yeah,
call
okay,
you.
ma'am. Okay, ma'am. Okay.
Okay, so shall I continue?
Yes, ma'am, please.
Is is it being repeated? Am I repeating whatever Pramod said? Raghu?
No, ma'am.
Okay. So I'll just tell you quickly about what reactive and preemptive testing is. Reactive testing occurs when patient is experiencing adverse effects which are not explained by drug-drug or drug-disease interactions, or when the use of a high-risk drug is anticipated and the patient's genotype is obtained much before therapy. And preemptive testing occurs when patients are screened for multiple pharmacogenomic variants prior to developing an indication for specific pharmacotherapy. And the development of patient individualized regimens should include assessing the patient's pharmacogenomic profiles in addition to the other information that we get, like adverse history, allergic history, adverse drug reaction history, any interactions, some dietary factors, and adherence patterns, etc. And there is a company called as CPIC, or Clinical Pharmacogenetics Implementation Consortium, and the that has published some therapeutic guidelines for multiple drug gene pairs. And also the FDA provides a list of drugs for which pharmacogenomic markers are included in labeling of drugs. So the role of the pharmacist in providing pharmacogenomic services: one is the knowledge and skills of clinical pharmacists, especially in pharmacokinetics and dynamics, gives them the advantage to take the lead and provide services in this area. And ASHP also has highlighted the ability of the clinical pharmacist to lead efforts to develop guidelines and protocols and to initiate pharmacovigilance services. And any clinical pharmacovigilance service should include these responsibilities, that is, promote a rational and routine use of PGX testing, that is pharmacogenomics testing. That is, it should not be done in random for every person, and it should be limited to those who you think are it should be a very judicial decision that should be taken by the doctor. And interpreting test results and communicating with patients and other healthcare professionals, and optimizing the therapy based on the test results, and providing information on the clinical application of genomics to healthcare professionals, patients, and the public, and contributing to research in this area and other platforms concerned with the integration and application of pharmacogenomics into clinical practice. And the responsibilities of a pharmacist for pharmacogenomics include promoting the optimal use and timing of pharmacogenomic tests. It's not that you start off a drug and then when the patient does not respond, you go back and do a
Sorry, madam, we cannot hear your voice. Hello? Madam, we cannot hear your voice. Respected participants, I think there is a slight network issue from Savita Madam's side. We request you to be in the program for a couple of minutes. Might be she will be joining in a couple of minutes again. Thank you.
Sorry for that.
Yes, ma'am, we can hear you now.
Yeah. So shall I continue?
Yes, ma'am, please.
So we were discussing the responsibilities of a pharmacist and to optimize the medication therapy and educate and provide information on the application of pharmacogenomics to health professionals, patients, and public, and to support research in this area and networks that guide and accelerate application of pharmacogenomics into clinical practice. And also, you can design a patient-specific drug and dosage regimen based on the genomic profile of the patient, and that should take into consideration the PK/PD properties of the drugs, and these factors should be combined along with other patient-specific factors. And another important responsibility of a pharmacist is documenting the interpretation of results in the patient's health record. And those pharmacists who have specialized education, training, or experience should have these additional functions of developing specific clinical decision support tools in electronic health record systems that will guide the prescribers on appropriate use and dosing of medications, and develop a process including patient-specific educational materials to explain to the patients the importance and significance of pharmacogenomic test results, and developing institutional guidelines and processes for implementation of a clinical genomic service, and to have a communication process for patient-specific results, and to document the same in the health record. And the gist is basically that you have to promote a collaborative relationship with the healthcare professional, and to encourage the development and appropriate use of pharmacogenomic principles in patient care. Since this is an evolving field and there's a lot of ethics involved in pharmacogenomics, it's very important that every process that is done is documented. And also, it is the duty of a pharmacist to contribute to the body of knowledge in pharmacogenomics by conducting and publishing research in that area. So I just have this study which was done where they taught a pharmacogenomic course to pharmacy students. So basically, there are 5 core competencies where pharmacogenomic education needs to concentrate, especially in pharmacists. And this has been given by the National Human Genome Research Institute. The first is terminology in genomics, and knowledge, then interpretation, and then communication and professionalism. So this Galvez Peralta and his colleagues have effectively managed to teach a pharmacogenomics course, and they published their findings in American Journal of Pharmaceutical Education. And so basically what they did was they designed a course titled Biopharmaceutics and Pharmacogenomics, and that was to be given for the 4th year PharmD students. And the course was a 4-credit course with 1 hour per week for team activity or quiz, and 3 lecture hours. And they designed 4 team activities pertaining to pharmacogenomics. So one was a paper that explored Mendelian disease. So Mendelian disease is any disease due to genetic variations. It could be mutations also. And the students was asked to write a paper on any choice of Mendelian disease of their own. And the objective was that the student becomes familiar with using databases, the pedigrees, and genetic terminology. So when they read about the disease and they try to understand the terms, then they have to go back and see what each of those terms mean. So the core genomic knowledge as described by the NHGRI, among them, terminology, knowledge, interpretation, and professionalism was targeted in this activity. And the educational outcomes, so they have something called as CAPE, where they list out the educational outcomes for every course. And if the student presented a paper or wrote a paper on Mendelian disease, then the knowledge, the outcome was that knowledge was about that disease was achieved. And the next activity that they planned was a verbal presentation on pharmacogenomics, that is any choice of drug of the student which had a genetic variation, and about the pharmacokinetics and pharmacodynamic effects of that variation, and whether any drug interactions were there, and other things. And that required them, the objective of that exercise was that they would know what is the level of evidence that is available for that drug to be given and for what kind of patients to be given. And they had to refer this database. I have a slide on the web resources, and so there's this farm GKB, and they had to refer that, and that database also gives dosing recommendations and what are the counseling points that has to be told to the patient. So the core genomic knowledge was terminology, knowledge, interpretation, professionalism, as well as communication, since they also were reading about what information to be given to the patients. And the outcomes, the educational outcomes that they were targeting in this activity was knowledge and improving communication, and also developing empathy and self-awareness. And the third activity that they did was a genetic counseling session. So they called a genetic counselor who are professional genetic counselor, and they got a hands-on training with them about um, they were acting as patients, and the genetic counselor counseled them as a patient who had this particular uh, uh, abnormality, and how they were supposed to go about it. And then the students really went ahead and had another role play where they were the counselors and somebody else was acting as a patient, and they actually were giving the counseling to the patient. And when they did this counseling, they chose the same drug which they had a presentation on, or the paper where they had presented about a particular Mendelian disease. And so this activity improved their knowledge, interpretation, professionalism, as well as communication, because the role playing was there, and they had to get into the role of a genomic counselor. And apart from knowledge and empathy and self-awareness, it also improved their communication, because they had that experience of actually sitting in front of a patient and having to counsel them. And the fourth activity was a class debate on personalized medicine and genetic sequencing. And the objective was to make it known to them about the ethics and laws and all the regulations that govern pharmacogenomics. And they were also made to look into social media posts and other uh, uh, web literature, and to know what are the regulations that govern the general part of medicine, and another group was against the personalized medicine. And they it taught them to see both sides of a situation, and it gave them terminology, knowledge, interpretation, professionalism, as well as communication. And this activity was also had an outcome of understanding the cultural sensitivity. So like, for example, if a genetic testing had made a patient come to know that he is genetically vulnerable to get a disease like, say, breast cancer, so what will be the implications for that patient and his family? Like, what will they be going through, and what are all the cultural issues, especially if it's like a country like India, and a girl getting a diagnosis, getting to know that she's going to get breast cancer, even before marriage. So the marriage issues and those kind of things, all those things was also understood by the students. So they performed these four activities um, for the students to understand, increase their understanding of pharmacogenomics. And they tested the various levels given in the Bloom's taxonomy. And they had some questions for every part of Bloom's taxonomy. For knowledge, so they had more than 80% scores correct answers for knowledge, for comprehension, for analysis, for application, and also to synthesize, evaluate, and create. So the authors reported that there was an increased level of confidence, awareness, and knowledge among students to address pharmacogenomic cases with patients or with healthcare practitioners or healthcare providers. So these are the various web resources for pharmacogenomics. Um, this is a natural language this thing and manual curation variants. There was this database will tell association between variants and the drugs, and which are the drug centered pathways, and genotype based pharmacogenomic summaries. So all this can be accessed from this website. And also this CPIC or this consortium, you can get a detailed gene or drug clinical practice guidelines and drug dosing guidelines from this website. And a drug bank, so this is a drug based information and including genomics, drug food interactions, metabolic enzymes, even quantitative structure activity relationship, okay, and ADME data. So that can be accessed from this website. And then PACDB, you have pharmacology related information, including the various genotypes, the gene expressions, and pharmacological data obtained from from a lymphoblast cell line. So where they grow cell lines and they try to see what are all the different gene expressions and genotypes. And then you have the CYP450 P450 allele, where you can see which are all the different alleles and what are their isoforms and what is the relationship between the genotype and the phenotype. And then you have the CYP450 drug interaction table, where they have given detailed interactions for the isoforms of the enzymes. And also the FDA's pharmacogenetic website gives information regarding dosing recommendations according to genotypes, the mechanisms of the drugs, and the germ lines or the somatic gene variant biomarkers. And so these are some of the important web resources. So these two are really good resources to look into if you want more information. And the other related information, or if you want any further information, you can go for this. And if you want to conduct studies of cytochrome P450, then you would go look into this interactions as well as CYP450. And then you have this scan, this thing that's more for pure science researchers and pure science, where they are actually looking at the actual SNPs and genome wide association studies. So that is a GWAS tool. So coming to the end of my talk, I'll just summarize the key points. So literature shows that application of genomics in clinical practice will improve the efficacy and minimize the untoward effects of drugs. And future implementation models of genomics and precision pharmacotherapy will include integrating this information. And this advances will require health information technology and effective electronic health record systems, and data storage, and then mapping and integrating with electronic health records, because we have in India, especially where there are several small hospitals which mushroom daily, around the city. And how many of those hospitals have electronic health records, and how do we link all of them? And also the ethical, legal, and social concerns should be taken very seriously to avoid any discrimination against individuals based on their DNA. and more outcome studies are needed in order to quantify the impact on health outcomes. And advanced pharmacist functions in applying pharmacogenomics in clinical practice may require specialized education, training, and experience. And pharmacy teachers have the responsibility of training the students and contribute directly to improved healthcare. So these are my references.
Thank you, ma'am, for your valuable talk. Now the session is open for questions. The participants can unmute and ask their questions. And we have a few questions from the participants. Are clinical pharmacists hired in pharmacogenomic labs directly, or do we need research experience?
Maybe if you're at the starting level, it would help if you can undergo some genomic courses like certificate courses. There are several courses that are available. So while you're doing your 5th year or 6th year, if you can do those courses, then I think it should be an additional feather in the cap for you all, so that the employer chooses you over anybody else. Because there are people with hardcore genomics background, so they'll be doing BSc and they would have come with all the life sciences where they would have studied genomics as a subject in their study, in their courses, in the many years of their study. So if we want to beat them, then we need to be extra smart, and we need to have some extra education regarding that. So it would help if you can do some courses.
There's another question now. Are there any internship opportunities by National Institute of Health for carrying out research in pharmacogenomics?
I haven't actually looked at National Institutes of Health, but I think if you want to get into that, you must have done some research in that area before you can actually get this thing. And you should have a very good bio data if you are applying to NIH for this thing. But you can apply for a grant, but you have to state your objectives and purpose very clearly. Did that answer your question?
Yes, there's another question now. Is there any difference between electronic health records and clinical data management?
So electronic health records are generally in hospitals, like patients getting admitted and getting discharged. So hospitals which have an electronic health records have this. Okay. And EHRs can also be taken from insurance companies, where insurances have paid for person's hospital stay, and so there are many sources of EHR. And what was the other thing you said? Clinical data management. So in clinical data management, you have to look at databases which have only clinical trial data. Okay, so all the data which has come from a trial will be in that data, and then the data management committee's responsibility is to go through the data, analyze it, and put out the findings or whatever. So both of them are databases, but probably the clinical data will be more of the trial, the phase in which they have seen the patient, whereas EHRs will be mostly regular patients getting admitted. It can be taken from insurance companies, life insurance companies, also because if you want to do a study on how many deaths due to myocardial infarction or anything that was there, if you want to see how many insurances were paid, so that also becomes a data. So they are two different things, entities, but if you want to analyze any of those things, you have to approach that particular person. I hope this answered my question.
Yes, ma'am. Thank you so
Answered your
much,
question.
ma'am, for taking time out of your busy day for us. Now we'll have valedictory session for this one-week program, ma'am. I request our director, Dr. Kumar V.S. Nagani, to give valedictory note.
Yeah, good afternoon, everybody. So, am I audible? My voice is clear?
Yes.
So, good afternoon, everybody. And it's indeed a pleasure for STTP to organize such an event. This event, there are about 1,000 registrations, out of which about 800 are the faculty and 200 are the students. So it's a great registrations, and I believe most of you enjoyed the talks. The sessions that was designed are covering various topics, starting from therapeutic drug monitoring, pharmacogenetics, pharmacogenomics, the individualized therapy or personalized therapy, and more importantly, the recent trend or the future trend, which is artificial intelligence and machine learning. I think the topics, I must thank the coordinator of this session, which is the principal of this college, Dr. Natraj, and the co-coordinator, Dr. Papiraju, for organizing a wonderful event. The topics that the theme that is selected is so good, and uh, every speaker has done a great job. I got so busy during workouts, I could not attend, but the talks are so interesting. I watched most of the videos in the night. So I must thank all the speakers for making this event a grand success. And I believe all the participants also enjoyed the same way. And these videos are available on YouTube. And any of you are interested to watch again, or you want to recommend any your students, you can ask them to subscribe for the YouTube of Sri Vishnu College of Pharmacy and watch these videos again and again. And I also must thank our PharmD 50th year students for facilitating this process. They did a great job, and they did very lucidly and very easily also. I think it's a great exercise, and the SCCP team overall has done a great job. And it is you people have to give us the feedback, and we'll see that the certificates will be uh, reach your emails at the earliest. And I must once again congratulate all the participants who has done this event a great success, and also thank all the speakers who really made this event a great success. Any event is of this magnitude cannot be successful without the participants. And I saw a great interaction in today's session as well as in all the sessions. I think all of you really enjoyed it, and looking forward to organize such events in the near future. So thank you all. So with this, I will pass on to the facilitators for next steps.
Thank you, sir. And now I request Anil Kumar, sir, to deliver vote of thanks.
Good afternoon, all. So, as we are coming to the closer of the session, so I request any of the participants to please give your feedback on the sessions.
You may unmute yourself and give your feedback.
I request any of the participants who are in the session to please give your feedback. Okay, so on this note, I want to conclude with my vote of thanks on behalf of Sri Vishnu College of Pharmacy. First, would like to thank AICTE for sponsoring this one-week online STTP on clinical pharmacy services and optimizing patient health. And I would like to thank all our patrons. So particularly, so our chairman, sir, K. Vishnu Raj Garu, so Sri Ravichandran Rajgopal, vice chairman, so Sri Aditya Visram, secretary. So our director, Dr. Kumar V.S. Nemmani, and our principal, Dr. K.S. Nataraj sir, for their constant guidance and support in conducting this one-week event. And especially, I would like to thank all the guest speakers who made this event great success and a very reasonably good experience for all the listeners. So Dr. Pramil Tiwari, Mr. Satish, Dr. Krishna Undela, Dr. Survel Rajan, Ms. Savita, Ms. Ramesh Naidu, Dr. Sriharsha, Dr. Pramod, Mrs. Richa Goyal, Dr. Jagadish, Dr. Kanan Gopal, Dr. Pravalika for their inputs and sharing their valuable time and also their knowledge with all the listeners. Thank you all the speakers on this note. We would like to thank all the student coordinators, Anusha, Mautika, Leela Amrita, Padmini, Pragnya, Meghala, Manikanta, Harsha, Arjun, Dimple, Rupa, Appal Naidu, Ashank and Nuruddin for their timely help and also their involvement in these sessions. We would like to thank Mr. Prashan Raju and also Vinod Kumar Raju for their help in technical session. We would like to thank all the faculty, staff, and students of SVCP in making this 7-day event a very successful and a happy learning to all the listeners. We particularly want to thank all the participants. Hope you had a great learning experience throughout this weekend, and thank you for participating. We wish the same type of encouragement and involvement of all the participants in further events. Thank you.
That's all for today's session. Thank you all. Thank you all. Very soon we'll come up with new STTP from our college, and hopefully we all have your attention in that. Thank you all.
Feedback will be shared to you at the earliest, and also we have an assessment form which will be shared to you by evening to your mails. So I request everyone to submit that in order to process your certificates within 1 week. We'll process your certificates and it will be delivered to you. Thank you all.